Welcome to part two of the gastrointestinal infections and food poisoning. This is the first part of part two as we continue our discussion of gastrointestinal infections and agents of food poisoning, my very favorite topic. So in part two of this lecture presentation, we will briefly review the characteristics of each of these pathogens. And although the discussion is limited to the 7 listed here, don't overlook, or more than 7, I guess, about 9. Don't overlook, and therefore you should also be familiar with other significant pathogens, such as Helicobacter pylori and Clostridium difficile. I will briefly review the events caused by these two organisms, and because they're not routinely cultured in a clinical laboratory, our discussion would be minimal, essentially when to expect these particular pathogens. So this will be in part two of this presentation. So first, let's take a look at and remember our opening case study and do a case check. Our opening case study, or case in point at the beginning in part one of this lecture, was about a 32-year-old man who traveled to a small village in Mexico and experienced a sudden onset of diarrhea 4 days after his arrival. He complained of nausea and the stool was watery, evolved without grouse, blood, pus, or mucus, and he had no fever. So, in addition to the history of travel, so these are some of the points to consider that we talk about. So, in addition to the history of travel, the individual also presented no fever, there was no blood or pus or mucus in his stool. And these are indicators. So based on what we talked about in part one, first, what would be the pathogenic mechanism deployed by the offending organism? What information are you learning, and where would this lead you into the investigation and recommendations you would make to the clinician should you be the microbiologist working on this particular sample or this particular case. Regardless of what we think in all those parameters, diagnosis of acute bacterial diarrhea should include the culture of appropriate body fluids. So I suggest that you review the functions, observed morphology, and biochemical reactions observed on each of the routine primary media for stool cultures. Make sure that you understand the functions of each component. What carbohydrates are in McConkey, hectoin, what is XLD and what does it do, what does it tell you with regards to the colonies that you recover in this different highly selective medium. What reactions can you tell by observing the colony morphology? When there is black center, green with black center on hectoin, what does it mean? What about when it's yellow? How would you interpret those observations? Routine culture media such as those listed here are routinely used. So when you set up a stool culture, usually it would include blood agar, MAC, HE, XLD, and a CAMP agar. Now, other selective or special highly selective mediums such as TCPS, CCFA, and SMAC are added to the protocol when indicated depending on your geographic location where you live or depending on the situation. So let's do a review of the agents of diarrheal diseases, starting with Salmonella. We know that Salmonella species are transmitted by food or water and are found in a wide variety of animal hosts. Salmonella typhi, however, is an exclusively human pathogen. Meat, poultry, and eggs that are intrinsically contaminated by non-typhoidal salmonella are common sources of infection. Food handlers who are carriers transmit the infection in improperly cooked or prepared and or improperly refrigerated food and other food products. For example, Salmonella enteritidis in eggs survives in omelette and French toast. These are food that previously have been considered cooked and safe to eat as is, out of the box, if you may. Other food products such as lasagna and meringue pie have also been associated with Salmonella infections. As a review of the clinical infections caused by Salmonella, clinical infections of Salmonella species include typhoid, paratyphoid, and enteric fevers, gastroenteritis or food poisoning, bacteremia, and what we call a carrier state. We'll talk about typhoid fever and gastroenteritis in the next few slides, but I just want to briefly mention a couple of points here with regards to bacteremia and carrier state. Bacteremia may occur in about 4% of immunocompetent persons with Salmonella gastroenteritis. Unrecognized episodes of transient bacteremia are probably common, but more prolonged bacteremia correlates with immunosuppression. So knowing that 4% of immunocompetent persons may suffer from bacteremia in Salmonella gastroenteritis or food poisoning, when the bacteremia is prolonged, this may be an indicator for some form of unrecognized immunosuppression. Make sense? Okay. The asymptomatic carrier state develops in about 0.2% to 0.6% of persons with non-typhoidal salmonellosis. And 10% of typhoid carriers will shed the organism for at least 3 months. And some of them will develop into chronic carriers. And long-term carrier state of salmonella, finding salmonella in stool or urine cultures may even last for about a year or over a year. So you have to remember that the carrier state for salmonellosis may last up to even a year. It is more common in women and in persons with abnormalities of the biliary tract. Therefore, if you receive a sample of gallbladder or biofluids, do remember this. If you receive a sample of gallbladder or biofluids, make sure that you include culture media that would recover salmonella, and that is, you should specifically include Hectoin or XLD and look for salmonella because of your knowledge of the carrier state should you receive a gallbladder or biofluid sample in the clinical microbiology laboratory. So let's continue. This is a graphic representation of the culture and serologic diagnosis of typhoid fever, and I will explain this graph in just a few minutes. We know that typhoid fever or enteric fever is an acute systemic disease caused by Salmonella typhi. A transmission may occur person to person or by ingestion of food or water contaminated by the urine or feces of acute cases or of carriers. Common symptoms will include insidious onset of persistent fever, headache, malaise, anorexia, constipation more commonly than diarrhea, especially during the first week of the infection. And take note of this in the graph shown here where it shows that the illness evolves over several weeks and occurs in different phases or stages. And that matters when it comes to determining what clinical sample is best to recover the organism and when. So if you look at this graph, during the first week, on week 1, when there is a sustained fever, this is the best time to collect the blood culture. This is when blood cultures will render the organism positive, and your chances of finding the organism in the blood declines during week 3 and as the disease progresses. Meanwhile, if you look at this graph on week 1, that there's a low chance of finding the organism in the stool because, again, as we mentioned, that constipation, more diarrhea, is more common because the organism has not reached the intestinal tract. But if you look at the second week of the disease, the second and third week, this is when the patient experiences diarrhea, where the organisms may be isolated from the stool. During the second week of the disease, salmon-colored macules called rose spots, this is a hallmark of typhoid fever, will appear on the trunk and abdomen of the patients. This is as a result of the organisms present in the current bacteremia. So it's not until the third week or so that the patient experiences diarrhea. That's when the highest percentage of positive stool culture will occur. In severe cases, intestinal bleeding and perforation caused by erosions of the Peyer's patches will occur, and the organisms are sequestered in the gallbladder. Therefore, again, as I mentioned in the previous slide, if you receive a sample of gallbladder or bifluids, make sure that you include culture media that would recover salmonella. The long-term carrier state, as previously mentioned, and the stool will be positive for salmonella, stool and urine cultures even, for at least 1 year. If left untreated, it can be fatal in 10 to 20% of the cases. The incubation period is about 5 to 21 days after ingestion. And now there are antibiotics and vaccines available for typhoid fever. But the elimination of typhoid as a major public health problem in the United States was due largely to improved sanitation rather than to antibiotics or to the vaccine, which is only partially effective. There are still occasional outbreaks of typhoid fever, but about 3/4 of cases are acquired abroad, and especially from Mexico, the Philippines, and India. The risk of typhoid among U.S. travelers is greatest among those who visit the Indian subcontinent. Gastroenteritis caused by non-typhoidal salmonella strains is extremely common in the United States, with an estimated 0.8 to 3.7 million each year. And the serotypes commonly involved are what you see on this slide. Salmonella serotype Enteritidis vag type 4, PT4, and PT104 have been implicated in most cases, but certainly other serotypes have also been implicated with salmonella outbreaks. Gastrointestinal infection due to non-typhoidal salmonella usually presents as nausea, vomiting, and diarrhea 6 to 48 hours after the ingestion of contaminated food or water. Review the common food sources for salmonellosis: poultry, eggs, turkey, So remember me during November, Thanksgiving dinner, as you have your turkey dinner. That's a Thursday. Um, Symptoms starts between 6 to 48 hours and sometimes 72 up to 72 hours. So from Thursday to Saturday, look for news of outbreaks due to improperly cooked or prepared turkey dinner. A high inoculum of Salmonella correlates with increased severity and duration of the illness. So the more turkey you eat during the turkey dinner, the more likely you may experience the symptoms, which include stools that are usually loose, moderate volume, blood may be present, and as well as mucus. You'll have fever, chills, nausea, vomiting, and abdominal cramps are the common symptoms. So how do we recover the organism? To recover the organism from clinical samples, they should include a highly selective medium, such as hectoin or XLD. And I'm not going to spend time with this, as we already covered this in micro 1. Just make sure that you review the reactions taking place in each of these media. Why they appear green on hectoin with black centers. What does that indicate? The reaction seen on XLD. So make sure that you do review the composition of this highly selective medium, specifically for isolating enteric pathogens such as Salmonella. Biochemical features. Similarly, this is just a review of the typical biochemical features of Salmonella species. So how would you differentiate other H2S producing enteric species like Proteus from Salmonella? Salmonella will have a TSI reaction of alkaline over acid with H2S positive. But so that's Proteus. Citrate, depending on the subspecies or the serotype, but most will have a citrate positive reaction, although some may be negative. They are all modal, okay? LIA or lysine iron agar, most will decarboxylate lysine, so the reaction would be purple over purple with H2S because LIA has an H2S indicator. But some certain species may not decarboxylate lysine and therefore will produce a purple over Y reaction. But one thing that will eliminate or exclude Salmonella from other H2S producing enteric Enterobacteriaceae is if you see a plum or red over yellow on LIA, which means that the organism deaminated lysine instead of decarboxylate. And remember that the group of organisms that deaminate instead of decarboxylate are the Proteus providencia group. So remember that Proteus hydrolyzes urea and Salmonella does not. So if you see a positive urease reaction, you know you do not have Salmonella. Okay. Salmonella does not hydrolyze urea, and Salmonella does not deaminate phenylalanine or PAD. So Salmonella is PAD negative, Proteus is PAD positive, and those are some of the key biochemical features that you do need to review and remember. So let's move on to another enteric pathogen, and that is Shigella. Shigella species are the classic agents of dysentery, and they are highly communicable. Shigellosis remains an important cause of morbidity and mortality in developing countries, mainly in children. At least 19,000 cases still occur in the United States each year. As we know, Shigella are gram-negative bacilli with 4 recognized serogroups. Group A is Shigella dysentery. Group B, Shigella flexneri. Group C, Shigella boydii, and Group D, Shigella sonii. Make sure that you know which one goes with whom. So use a mnemonic as far as A, B, C, D, and what it corresponds to. Within these 4 serogroups are about 40 serotypes, of which Shigella dysentery serotype 1, also known as Shiga bacillus, causes the most severe disease. Shigella sonii, group D, Shigella, now accounts for about 60 to 80% of cases of shigellosis in the United States. Shigella species are strict human pathogens and are spread via fecal-oral spread in contaminated food and water. Flies and other insects carrying the organisms may spread the organisms. The infective dose for shigellosis is very low. Less than 100 organisms may initiate an infection. As little as 10 organisms actually may initiate an infection. So be very careful with this organism when you are working with it in the clinical laboratory. We had an incident in a hospital in San Antonio where I was visiting one of our clinical affiliates for our students in clinical rotation, where the technologist became infected with Shigella while working with the Shigella organism, and she was inoculating one of the test tubes, and she contracted Shigella. So be very careful. Symptoms include severe abdominal pain with cramping, diarrhea, fever, nausea, and vomiting. Feces will contain white blood cells, red blood cells, and mucus. So although the organism does not reach the bloodstream, this is unlike salmonellosis, where the organisms will cause bacteremia. Shigella does not. It produces, however, various toxins, like the Shiga toxin, that can have cytotoxic effects in the intestinal mucosa. Hence, the white blood cells, red blood cells, and mucus. It is very destructive in localized invasion in the intestinal tract. So, identification of Shigella species. Again, on McConkey, it will produce clear colonies because they are non-lactose fermenters. They will appear green on HE. And XLD will produce red colonies. They do not produce H2S, and therefore there will be no black centers as compared to Salmonella colonies. Biochemical features. This is one of those that I tell students, this doesn't do anything. So remember, everything is negative with Shigella. It does not ferment lactose or sucrose for this particular Shigella species. So you will have an alkaline over acid, no gas, no H2S. Citrate is negative, motility is negative, everything is negative. Okay? So review the characteristics and biochemical reactions and make sure that you are able to identify Shigella species on the biochemical set of TSI, citrate, urea is negative, SIM, that is SIM, sulfide indole motility, is all negative. It is non-modal. Next, let's talk about Campylobacter jejuni. Actually, it's the most common cause of bacterial diarrheas. This is a zoonotic pathogen. And in the United States, infections due to Campylobacter have ranked the most common cause of diarrheal infection in the United States. And it is isolated from stool cultures more frequently than either Salmonella or Shigella species. So you will likely encounter Campylobacter during your clinical rotation. This is found in a wide variety of animal species, and outbreaks occur from improperly prepared poultry, untreated water, and unpasteurized milk. Human disease is predominantly foodborne but can result from direct contact with animals, including household pets, like your cute puppies or kittens with diarrhea. Fecal-oral transmission occurs, and men who have sex with men are at increased risk. And this is also true with Shigella, where it has been associated as a sexually transmitted illness in this population. Antimicrobial resistance has been recognized in Campylobacteriosis. Campylobacter isolated from human infections have become more resistant to antimicrobials, particularly with fluoroquinolones, after these agents were introduced for use in animals. This is just a graph of interest where Campylobacter jejuni, it shows how it causes disease throughout the year in the United States, but sharp peaks of Campylobacter disease occur during the summer and early fall. And this graph also shows the affected population. The disease especially affects young children less than 1 year of age and persons between 15 and 29 years of age. There was a report some time ago that is the frequency of infection is seen more often among young adults, especially in men. And that is, this is the time when they leave home. You know, if you look at this graph, between 20 to 30 years old, there's an increase in both Campylobacter and Salmonella cases. This is about the time when they leave home and they start their own apartment living, perhaps cooking, or just by living by themselves. Well, I just thought I'd just throw it in. Again, this graph is just a matter of interest. Recovery and identification of Campylobacter jejuni in the laboratory should include a selected medium such as the Campi agar. Environmental requirements include 42 degrees centigrade incubation, and the colonies will appear moist and runny. And if you look at and it should be in a microaerophilic environment. The gram stain will show this seagull wing-shaped morphology, characteristic morphology, because they are gram-negative curved or spiral-shaped rods. It has a characteristic, what we call a darting motility on wet mount preparation. Now, because Campylobacter is also oxidase positive, just like Pseudomonas aeruginosa, and Pseudomonas aeruginosa will survive at 42 degrees incubation, the same environmental conditions. Make sure that you confirm your isolate with recommended biochemical tests, and one way to differentiate if you have a Pseudomonas or Campylobacter, is doing a gram stain or performing a wet mount preparation, observing for darting motility or the characteristic morphology of Campylobacter. This ends part 2.1, the first part of reviewing the characteristics of the agents of diarrheal diseases, and we'll continue with part 2.2.